Thank you very much. I would also like to thank very much the organizers for inviting me here to this really very exciting conference. Now, what I want to talk about is applications of probabilities to evolution, and I was successful this time. The talk is already on my webpage. If you are interested, you're welcome to download it from here. This is a kind of menu of my talk. There are 3 sections which are actually dating back to some time ago. Before I come then to the major part, evolution and so on. Now you see there are 2 provocative questions already. Is evolution possible? And the second question is non-probabilities. Perhaps 10 years ago, I would not have added these 2 sections. However, developments that intelligent design work and creationists coming up in the United States and in Turkey, evolution is no longer taught in school. So I thought I put these 2 short introductory comments into it. And so let's start with the question, is evolution possible? And I would like to quote 2 famous scholars who did the same, and in a provocative way, I may say the same mistake in estimating probabilities. Both comment on assembling elaborate structures with specific functions through random events and claim it's impossible. And I will show you why. A famous Eugene Wigner was referring to John von Neumann's self-reproducing automata, and he was of the opinion that it's impossible that you can, by random events, get such an elaborate structure. Fred Hoyle was much more pronounced. He was talking about bacteria, and he claimed evolution is impossible. The bacteria must have come from somewhere in space and come to the Earth in that way. Now, the statement above is used as an argument against Darwinian evolution, and in this context also the terrestrial origin of life. This argument is neither correct nor incorrect as long as it's not clearly said what's meant by random. And I give you here 3 examples. We have well-known different degrees of randomness. Random as in random numbers, and that's how the calculations were made by these 2 people. Random as in random walks. That's quite different from random numbers, because in a random walk to a random number is like a Bernoulli process to a Markov process. Random walk has always a point where from step to step it proceeds, whereas Bernoulli numbers always start from scratch. And the last example I have here is targeted random paths. That's something that people do often in excursions. They start from somewhere and they meet at a restaurant. And the path in between is random. And you will immediately see that this degree, these 3 different notions of randoms are very different. Random numbers are, you may say, complete randomness. In random walks, this is already reduced in a kind of progression of the Walker, and in targeted random paths, I even have fixed the final point. Now I give you the Fred Hoyle's bacterium calculation. So what he says, this is his starting point: all genomes have equal probability, and all except one have no survival value. They are all lethal. And then you may take the size of a typical bacterium, a small bacterium with 1 million bases and nucleotides in the DNA. The alphabet size is 4. Then you have 1 million nucleotides. The number of possible genomes is 4 to 1 million. And so the probability to find a given bacterial genome by a kind of lottery is 4 to 1 million. This is 10 to minus 600,000, and I think I have indicated here this 600,000 zeros in between. And here are the two references. Fred Hoyle, The Intelligent Universe, is a whole book he has written, and this is part of this book what I was quoting here. What's the meaning of such probabilities? And I'm a little bit provocative in saying these are non-probabilities, because imagine our Austrian lottery, 6 out of 45. So here you have a score of one particular event, and you can easily calculate the probability. This probability is that you get all 6 correct is 1 in 8 million. Now assume, let's say, 100,000 people are buying in the lottery. Then what you get is it's about once in 10 years, there will be a 6 hit. So you can easily guess what's the fate of this lottery. Nobody will play anymore if it's not even sure that he will survive long enough to get only to be possible, that it's possible that he observes one of these special gains. Now, with respect to the Austrian lottery, the situation is, of course, much better, because the maximum number of tips had been 52 million. This is the date of it. And usually, for an ordinary week, they have about 20 million, 10 to 20 million tips. So that the chance that you can observe a favorable event, that one person gets a sick, that is given. You can see these persons, you learn about this person, and you will play. If you are willing to waste your money on that, but it's something that you can observe. And so I think the important thing here is that one over this, this probability is in a range where at least one of events occurs in the time of observation. And this rules out all the probabilities we had before, because there are many times the age of the universe, and many times nothing happens. So nobody, actually, if this is a lottery, would be interested. And in the lottery, we have the random number situation of maximum randomness. Now, I will, it was already mentioned in a much more elaborate way in the previous talk, protein folding. And I call this an almost solved example, and it started out with a similar argument as the Wigner-Hoyle argument by Cyrus Levinthal. So what's the problem? I have an amino acid sequence. I look at a small protein, 129 amino acid residues. So here, this is the sequence in a code where you use one letter for every amino acid. And this molecule has a beautiful three-dimensional molecular structure. And Levinthal's problem actually was, I have this sequence and I can easily make conditions where this sequence is a random coil. So there are no interactions within the protein of non-covalent type. It's just an always moving chain, and this chain has to find the native structure. This is the native structure as kind of a minimum, finding the minimum free energy problem. Now let us treat this problem as we have previously seen with the question of evolution. The average number of conformations is perhaps a little bit too large with 8, but that's what Levinthal assumes. Plays no role when you put 6 or 5 in. Its argument is the same. The chain length is 129 amino acid residues. The number of possible conformations is now 128. So this is 0.4, 10 to the 116. And to find the probability to find this native conformation by a completely random search, so Bernoulli trials, like random numbers, this probability is 10 to the minus 116. And as I said quite previously, this is what I would call a non-probability for the simple reason, if you test 10 to the 13 conformations per second. And this is really quick search, but it happens with certain proteins in solution. It would take 1.3 times 10 to the 95 years to complete the search. But, and this is Levinthal's paradox, the protein change in nature can fold in about a second and finds this structure. And now, in this case, we have the solution. You have heard about proteins and how you, with very elaborate experiments, you can really investigate all what's happening there. Now, this is work that has been nicely illustrated by Ken Dill, but also other people before have worked this out. And so the probability as we calculated it is, one could say, a golf course problem. So you have a two-dimensional surface, a two-dimensional plane, and you have a ball rolling here, and you wait until the ball hits this hole, and then falls into the hole, and this is what we have just calculated, 1095 years. Now, Levinthal himself was suggesting that could be different. There could be kind of a path along which the ball may roll into the hole, and here it's much easier because you have to hit this path, and then you roll into the hole. So this is what has been called the pathway solution. But physicochemists, they were not happy with this solution. They said, why not considering here a funnel? So we have, instead of this plane surface, we have a surface where from every point you can start and will fall in here. And that makes it, of course, easy because you just start anywhere on that surface and you roll into the... Now, proteins are not like that. They are more complicated, and that you see it here. See here, this is structured. That means when you start to roll in here, then there are various possibilities. You get caught by some intermediate points until you fall in here. So this is Martin Kaplu's view of precisely this protein, the lysosome. And he picks out a very quick pass, then a slower pass, which has to go along a second saddle point. And then there is a third pass, which takes even longer. And this is what you find experimentally. You have a distribution of these folding times. So in 95, Pringles and Onosich, Sochi, and Peter Wolines. Peter Wolines was the driving force of this development, this kind of funnel theory for folding proteins has been developed. And the width here, which is the width of the funnel, this is related to the entropy of the process, and then you get immediates, and in those days only one intermediate was calculated. This was postulated, so-called molten globule, where you have partial structure already. So, but there is a big question mark. Biological landscapes for biopolymer folding or for evolution are high-dimensional and much more complex. So the picture is a nice illustration that I have given you with this two-dimensional surface, finding different paths where you can fall into the hole. However, situation should be more complicated. And protein and nucleotide folding landscapes can be investigated by experiment. You have seen beautiful example in the previous talk. And evolution can be studied under controlled laboratory conditions that provide insight into the mechanisms of biological evolution. I just want to show you, in case of proteins, you can study the Newtonian dynamics. You get the information that this is not a static problem, as it was already mentioned. This is a dynamical problem, and as such more complicated. And then what you need, and this is why I said an almost solved problem, you need a possibility to get information on this energy landscape, and this by Martin Kaplus, who won the Nobel Prize, I think, 4 years ago. He started out, and this is a part which is here related to what we know from quantum mechanics, bond energies, angles, and then you get dihedral angles. Then you have to add additional parameters, and some are called the impropers, which sounds a little bit funny. You have to add an additional force field, and then you have to add for the non-bonded pairs quite an elaborate function. But with this force field, you can calculate dynamics in the short range, and you see that this picture from 2 dimensions can be in a way generalized. So this is, now that was the excursion into the history about probabilities, and I would say how not to calculate probabilities without knowing the process. just assuming that a lottery is used for determining the next conformation that gives you usually wrong results. Now, I start very, very long ago about evolution, and there were two people, Robert Malthus and Leonhard Euler, who Malthus was using the observation that animals, also plants, proliferate like a geometric progression. So if you start with 1, 2, 4, 8, and so on, and Euler introduced for that purpose the exponential functions. Now this is nothing for you here, for this audience, but biologists always believe that a geometric progression and an exponential functions are things which are completely different. Now this I show for this purpose. The exponential function just is a kind of perfect interpolation between the discrete values of the geometric progression. So what the problem is, and this Malthus put this very clearly, whenever you have resources, enough resources, a population will grow by a geometric progression and will consume all the resources, because you can see that goes up and up and up. And so the only solution to that problem, mathematical solution, was given in 1838 by Verhulst. And Verhulst had the idea, I have a population, and the consequence of finite resources is, here I have, this is the part that gives you the exponential growth, and then he was adding a term where you have a quadratic term in x, and then some constant down here. And this quadratic term in x was taken from mechanical evidence. If you have friction, the simplest way to account for friction is you put in a quadratic term. And that actually, it's easy to solve, and that actually gives the solution to the problem. Here you have the exponential growth would consume any resources, but to be realistic, a certain ecosystem can only have a carrying capacity, a finite one. And if you do this, you have here this typical logistic function, logistic growth function. I was worrying, where comes the term logistic from? And Verhulst himself has used it already. And then I was looking in the literature whether somebody can explain why it's called logistic. And I found a very nice quotation saying, you have names in mathematics and physics and so on. They tell you something. They tell us something about the problem and about the kind of the solution which is in this. But there are other names that tell you nothing. And logistic is one of these. So don't worry, use it, and it's in common use. But forget about logistics and so on. This is something very different. And there's something which I found really remarkable, because what I show you now is everything could be done 20 years before Darwin's Origin of Species. There is no difficult mathematics in it. Yeah, I just want to, the basic structure of this equation is a term, a growth term, and a quadratic term compensating the growth and giving rise to a finite value. You can make chemical models for that, and this is necessary when you translate it in stochastics. It's much easier to have a stochastic model for the logistic equation when you can convert it into a chemical. And I have two chemical models here. The one is just the reversible autocatalytic reactions gives you this kind of growth term, exactly like what we had before. But we have another chemical model where you have two eggs annihilating. So this is not going to an equilibrium. This is just having which is practically the same because you can use here h/2 instead of h, and you have the same thing. Now, these two models are entirely different. One is useful, the other one not. And you have an absorbing barrier at x equals 0. We will see this in a second again. That means if all x are gone, since the basic reaction producing x is x plus a gives 2x, dx over dt equals 0. And this is an absorbing barrier. You can show it in, you look at the stochastic process and you can show that. And the annihilation reaction, which gives you practically the same differential equation, but when you go to the stochastic problem, you see immediately that this annihilation reaction here allows you to go from 2 to 0. So here you go from 1 to 2, 2 to 0, or to 3, and so on. And here, you cannot go to 0 because you need 2x to get 1x, and 1x cannot enter this equation anymore. So here, in case of this reversible autocatalytic reaction, you have a reflecting barrier. We know populations die out in nature, so when we use a model, we have to use this model and not this one. Oh, and now what you see here is the stochastic treatment. Here you have the mean value, and this is plus minus sigma, so one standard deviation. And you see here the deterministic solution. And this is very characteristic. The deterministic solution is faster. And this you find for all autocatalytic processes, for all reproduction processes. Now, two weeks ago, I have been here at a meeting and I have spoken a lot about this, what I would like to call stochastic delay. So, and now we can see we have here two states in the stochastic problem. This is the state of saturation where you consume your resources, and this is extinction. And one or two initial particles give you the same value, almost the same value for extinction, and so on. So we have, in the deterministic model, we have only the state S1. In the stochastic model, we have also extinction. And this will should be shown in a moment where it's important with respect to selection of the fittest. We can, and this is what I wanted to address already previously, we can generalize the logistic equation to a non-homogeneous population, to a heterogeneous population, where I have different x's, 1 to n. This is the concentration of x. And then, when I put this into the Verhulst equation, I get this equation, which is nothing but Darwin's natural selection, formulated in mathematical terms. So the logistic equation was there. And the only missing point was I don't have a homogeneous population. I have a heterogeneous population where I have subspecies or variants with different fitness values, and I get for granted. I get Darwinian selection just by it's a simple small transformation of the equation. For me, it's a mystery that the biologist hasn't done this 20 years before the origin of species. I have asked population geneticists and also other biologists whether I have overlooked something, because this is so trivial and so simple. And you get something else. You get for free the optimization problem, survival of the fittest, because you can show that this term here, which is the mean fitness of the population, this term, the time derivative is 2 times the variance of f, and a variance is always positive or at the minimum 0. So until you have the maximum fitness, you will always increase, and this is, in mathematical terms, survival of the fittest. I think here I have some integrations of these equations that are trivial. So red is the subspecies which has the highest fitness value, and at some times in the beginning, this species is selected. Now, in order to make it more general to analyze, we put it in a flow reactor where we have a defined environment. We let the resources flow into the reactor and the reaction mixture out of the reactor. And then we have a certain residence time of the molecules in here. Now we have a population. Let's say we have n. Here we have 5 different variants in the population. Then these are the corresponding kinetic equations, nothing special. And then here you get the differential equations, and trivially to solve, and what you get is what I have shown you already in the previous. You get selection in this flow reactor. All species except the one with the highest fitness values, and this is the red one. All the others are diluted out of the reactor. Now we want to do this in a stochastic way, and here we, instead of our concentration variables, we have now probabilities, and of course these are integer values because molecules, numbers of molecules can only be integers. And then I just show you the equation. It doesn't look very beautiful because here this is for A, where the discrete variable is m, and so you have, let me say I have 1,000 molecules of A, then you have 1,000 equations of this time. To solve, there are ways to solve master equations, but not in this case here. In this case, you are completely hopeless. So what can one do? And this is what we have done is you go back in the literature, and there is from Kolmogorov, Andrei Kolmogorov is the first hint how you can, through sampling of trajectories, approach chemical master equations. And then many people have done pioneering work in this field until in 1977, Daniel Gillespie wrote a routine which is efficient, easy to use, and this was used by chemists and biologists. And this allows you now to do simulations for these stochastic equations, as I've shown you before. And And this is a very nice proof that was done by Gillespie. In the limit of an infinite number of trajectories, the distribution of the trajectory bundle converges to the probability distribution of the corresponding solution of the master equation. So you have a numerical procedure, but you know, and it's proven, that this numerical procedure converges to a solution of the master equation. Now here I would like to make a commercial in my talk, in my book, 2 years out nowadays, and it has started from 100 pages notes of a lecture and ended up 750 pages. It's really a monster, and I have learned something that Phil Anderson said long ago. He said, more is different. And here I learned to make index register and so on for 750 pages is different than you make it for 100 pages. So now I look on various trajectories. Now this is one solution that you get, and they are all under identical conditions except the seeds for the random number generator. That's the only thing that differs from one picture to the other. Now this we know is the absorbing state of the stochastic process, because if all the x are gone, no x comes back, and it stays with all A, which is here in black color. Now, this is the deterministic solution. You get it in certain trajectories from the stochastic approach. You have a certain concentration of A, and here you have in red these fluctuations. And the values are very close to the deterministic value when you integrate the differential equation. But that's not all what I get. I get also the same process selecting x2. And x2 is not the fittest variant. It's the fittest but one. And if you are patient enough, you get also a selection of the least fit variant. In this case, I have only 3. This is number 3. It's easy to understand. I started from 1 particle each, and if in the first process 1 or 2 is diluted out, then 3 remains. Or if all 3 are diluted out, only. So the stochastic process gives you not only the deterministic solution, which is a quasi-stationary state in this case, it gives you also the absorbing barrier and these 2 as well. So now it becomes interesting when you try to do this as quantitative as possible, and you see immediately that if I have delta F is to F is 0, that means all fitness values are the same, then what you get here for 2 different population sizes, you get an equal distribution. And that's what Moto Kimura's neutral evolution is about. So the interesting thing is you get selection. Here you get 35% selection of I1, 34% of I2. So everyone can be selected. Since there is no fitness difference, the probability to select 1, 2, or 3, they are all the same. And I will show you a picture, how this can happen. It's very, very simple. What you do is you have 16 different colors. And this is a so-called Moran model. You simulate evolution by choosing 2 of these balls arbitrarily. One is multiplied, one is replicated, the other one is kicked out. So here the yellow is kicked out, and the blue one is replicated. and so on. And what you see after this is 11 steps of replication, out of these 16 colors, only 4 have remained. All the others were kicked out, and since they never come back into the system, you get here ultimately selection. If you wait long enough, you get. And this is what Mota Kimura called neutral evolution, or I would rather call it random selection, because the choice which of the variants is selected is arbitrary. Now you might say, why should one worry about such small numbers, 1, 2, 3, and so on? Now consider mechanisms in biology. In evolution, every mutant is created by a single copy. It either survives or it gets extinct. And this is the reason why whenever you do, I have to speed up. Whenever you do biological models, you have this to keep in mind. Now here you see this is we start with an empty reactor, and independently of the initial conditions and so on, you can always distinguish several phases of this process. I will be now a little bit more, a little bit faster. Now, we have 4 classes of fluctuations. And this is something that you get out from looking on these stochastic trajectories. There are the thermal fluctuations. They are in every chemical reactions. You find, and they follow this square root n law for the standard deviation. Then the stochastic delay, this is systematic, has to do with autocatalytic reaction. Occurs only with autocatalytic reaction, but of course also with multiplication. Then the so-called anomalous fluctuations. This is whenever you have bistability. You get a distribution of the 2 stable states. And this distribution that causes sometimes enormous additional fluctuations. And the last one I will discuss then there is just a few minutes. Thermal fluctuations, as I said, stochastic delay, and anomalous fluctuations were discussed in the 1980s already by Paolo Dombesi and his co-workers. So I think I will be very quick now here. The question is, we have seen these surfaces, these free energy surfaces for proteins. You can model it. It's a complicated functions, but you can in principle derive a model for it and have an analytical expression. It's not very accurate. It doesn't give you the experimental minimum, but something close to it. Now, how to proceed with RNA molecules? And there are 2 features which are very important if you have an RNA molecule. Whenever you make a mutant, and here what I do here is I change from A to U. At this position, I get a different minimum free energy structure. When I change from A to G, at this position, I don't change the structure. And when I change from A to C, I get a completely different structure. So, same position. I just change, make a mutation to a different base. I get, in one case, the same structure and very different structure. Different structure, here very different structure. The other cases. Now this is just looking at all possible mutation. And here you see as an example of a quantity which is derived from this structure, what this energy surface is. Here it's a free energy surface, fitness surfaces look the same. You have a certain percentage of neutral neighbors. These are all the neighbors of this sequence. Then you have some with very, very low fitness. This is practically very low energy. This is practically unstable. And then you have some which have higher free energy. And they are all in the one error neighborhood. And if you look now to the whole structure, what you see, I think I will skip this. No, this is something I need. What you see here is you have many sequences giving rise to the same structure, and they form something which we have called a neutral network. This is a network of sequences in sequence space that fold into the same structure. And remember, at the same time where we had this these structures with equal energy, we had very different ones, all in the same neighborhood, some the same. And these are about 30% are neutral. This goes through most biopolymers, goes also for proteins, about 30% are neutral. So from what we see here is we have this ruggedness and we have the existence of neutral networks. And these results are very general. They are true for other polymers, for example, for proteins. And then this evidence for ruggedness and neutrality and the existence of neutral networks that was obtained also from virus evolution and in vitro experiments with bacteria. So this we can skip. Yeah, and now this is the last part, the one I want to show you, the interplay of random drift and adaptation, adaptive evolution. So drifting randomly on the neutral network and then going in the sequence space up to higher fitness values. So, and these are the experimental system where this has been verified. Evolution in silico, well, you may call it non-experimental, but it's very similar to doing these in vitro experiments on evolution with RNA molecule. A lot has been done, virus evolution and bacterial evolution. Again, the flow reactor, then this is the way we have done this simulation. We have started from some arbitrary sequence giving this kind of structure. And this is our target structure. And the fitness is calculated with respect to the distance of the closer you are to the target structure, the more fitness the sequence has. And there is one generic picture what you get, whether you look at bacterial evolution, virus evolution, or this in silico. The approach is not in a way smooth. It goes in plateaus and fast adaptations. And here you finally have reached the final value. Now, this is the width of the distribution. And what you see is whenever the system is in an adaptive phase, you see an increase before this adaptive phase starts in the population width. Then it goes down. Increase in the population width. Then it goes down. Here you have a large increase. You have a long period here, and here the same. And this is the drift of the population center. So I look at the population center and how it moves in sequence space. And in general, it moves very smoothly and not very much, very slowly. But then, whenever you get such an adaptive phase, you have a peak. And this peak here goes up to this high value. This is a particular pronounced peak. So how can this happen? And this is just a picture here. I have the first loop with 3 arms, and this is one of these points where you get a major step. And here you have 2 of these loops which need only to merge, and then you get the final structure. So the characteristic features that we found in this simple simulation is interior loops with 2, 3, and 4 arms are key structures. And now I want to show you the spreading of a population and the collapse afterwards. And so I have my population, and then I calculate the two eigenvalues, which are the 2 largest. So the plane, which is spanned by the 2 eigenvectors, is the one which is the most characteristic for the population. And this center here, this is constant for the whole simulation. So this is put into the center. So now we see it starts spreading, spreads enormously, and then becomes smaller, and moves to another point here. So the population starts from the center here, spreads, and then all at once converges to another point. And this is what we have seen in the width of the population. We may go back here. Yeah, see here, in the width of the population, for every of these jumps, the population has spread, and then immediately jumps, spread, jump. And this is the position of the center. Here, the migration of the center. Here, very slow migration. Then a jump goes up to here. Then here is an enormous jump up to here, and so on. So, and this is what actually this movie shows. If you look down now, you see that the time is not homogeneous, so that this quick collapse is also reflected here by the time scale. So, okay, now this is the interpretation. We have a neutral network here, and the population more or less drifts randomly at this neutral network and spreads. And then we come to a point where it goes to the next neutral network, and come to a point where it goes to the next neutral network, spreads again, and so on. And we are now in a position to look what happens at this point, where it goes from one neutral network to the other. And it turns out that these are points where the probability to make a mutation that goes from one network to the other is sufficiently small that the population can cover it. And then I come back here to my beginning, what I said at the beginning. The probabilities have to be such that an interesting event can happen. And this is only here. When you calculate the probability to go from here to here on other points, it's much smaller. And this is the structure of these networks and the structures which are there. So I think this is the take-home lesson, how evolution on these neutral networks works. And then I give you another sketch, which again is a low-dimensional sketch, and we have to be careful with interpretations, but we see one feature very well. Now, this is what the population can do on this network. The size allows to bridge small values. You see from here to here, from here. But the population cannot bridge large values because it's not large enough. And how does it happen that it can do that? Now, there is a kind of tunnel where it goes from here to here. But of course, there is no tunnel in the landscape. But high dimensionality allows you to make random drift here on a neutral network until you come here. And this is, in a way, the trick that nature uses, going from one neutral network to the next until the target is reached. And here I'm optimistic, so I have drawn it in such a way that the target is actually reached. Now, 2 points which I think are important. Reproduction leads to selection. In case of no effective fitness difference, the selected variant is chosen at random. And this is something we have to keep in mind. There are all these philosophical questions of selection of the fittest. This is only true in large population. When you have a smaller population, you may select the next fit variant, the next but one, and so on. This is one thing. And the other thing is, for efficient evolution on natural fitness landscape, we require 2 things: adaptive period of fast fitness increasing, and they are accompanied by phenotypic change and periods of phenotype stasis with random drift in genotype space. So I've spoken too long. Thank you for your attention.